Hello, good morning, colleagues, to the ones here in the room and to the ones connected online. I'm Iris Mangas. I'm a regulatory toxicologist, and I have the pleasure of managing EFSA neurotoxicity projects and working with Brain Health Consortium. Today is an exciting day for us because we have here a huge amount of the researchers of Brain Health Consortium, and they will present the results so far on his projects on advancing NAMs for neurotoxicity. When Odvar told me we need to go to EFSA to present the half of the project meeting results, I told him it would be fantastic if we can organize an informal discussion also with our KIC, that is Knowledge Innovation Community of NAMS, and our IRMA community, that are two NAMS knowledge sharing communities in EFSA. I have to thank you very much, especially Martina from the Pesticides Peer Review Unit, Jochen and Sofia, also Martina for the IRMA project, and Alexia that is here with me. They have been in share or organizing this session. They will also help me recording this session, and this will be disseminated in EFSA, but also externally within the different social media. Today, I have also the difficult task of chairing this meeting. I say difficult because I am sure after these 2 hours, all of you will be very enthusiastic and you will have a lot of questions and you would like to know more about this topic. Good news is that we will have brain health colleagues with us until the end of the day today, and also that at the end of the project in end of 2027, we will have a workshop where we will discuss in details the results of the project. Jean-Luc will support with the Q&A and the final conclusions and open exchange of the meeting. So brain health is one of the 25 projects of IRMA, which is Innovative Risk Assessment Methodology. This is a program with a 51 million of budget of EFSA that aims to ensure we are ready for the future risk assessment, so ensures preparedness and up-to-date scientific approaches. This specific project contributes to deliver improve of risk assessment preparedness and increase preparedness for future regulatory needs. From this, we have 2 neurotoxicity projects as part of IRMA, and more or less 15% of the budget is dedicated to that endpoint. I have to say that the EFSA history of working with neurotoxicity NAMs, and in particular with developmental neurotoxicity in vitro methods, it has a longer history, and actually started in 2013 when our plant protection products told us, EFSA regulatory toxicologists, that we should do an effort in order to align the regulatory methods that we use for risk assessment of pesticides with the state of the science of in vitro methods and neurotoxicology. And since then, as you can see here, we have organized a series of workshops. We have worked in collaboration with OECD, and there are more than 10 EFSA publications on this area. We have also worked with OECD. There is currently an OECD DNT in vitro battery initial recommendation document. We organized a workshop, and we also have EFSA AOPs OECD endorsed on this topic. I have to say that since IRMA started and brain health started, there has been an increased amount of research projects on NAMs for neurotoxicity. We have here some of them, as SPARC, the EFSA One's Brain Health, or DNT RAP. So the knowledge on this topic is going to increase enormously in the next year, as you will see. Another important thing of the project for us regulatory toxicologists working on regulation is that from the research point of view, when this project started in 2005 by researchers, but also from the regulatory point of view, the question was never whether the NAMs can completely replace the current DNT paradigm, based mainly on in vivo studies. The question was more how these NAMs can be useful in refining and reducing the animal testing and producing, very importantly, more informative risk assessment for DNT. Currently ongoing, we have three main projects or three main actions in EFSA aiming to develop NAMs for neurotoxicity and aiming to implement this knowledge in the EFSA risk assessment. One of them is the brain health, and we will see today how much this project has advanced. It started in 2024 and will be running for 2 more years, and it has the aim to address some of the scientific uncertainties of the NAMs that we consider them ready today, which is the so-called DNT in vitro battery. It has also the aim of expanding the applicability domain, as could be to other contexts of use in EFSA, but also externally for other chemicals, for example, or contaminants. And it has the aim of refining the current available NAMs DNT testing strategy. There is another important area for us that is also another project of IRMA, and it is actually 6 projects englobed in the environmental neurotoxicants project. And this, it has a more regulatory implementation focus, as it aims to generate data and use the data in the current DNT in vitro battery. And also it aims to produce transferability, reproducibility, moving towards validation of the available methods. In addition, we have a series of activities in the pesticides peer review unit, mainly aiming to produce guidance and databases when we consider that the methods are sufficiently reliable and sufficiently relevant for us to implement this knowledge in the pesticides risk assessment. We have some examples as the development of a DNT reference chemical list that can be used for in vivo, in vitro comparison and performance analysis of the NAMs. Or we have an example very recently developed by the PPR panel on PBK guidance for DNT in vitro battery. We do all of this with our EFSA neurotoxicity working group, that is a scientific advisory board that supports us in the development of the projects. And I think here we have created, and maybe you can tell us if it's true, but I think we have created like a very win-win scenario where you researchers benefit that we have this scientific advisory board, and for example, we can discuss and select the most appropriate chemicals for your NAMs. So in a way, we are sure that the results of that will be useful for us. And from the other side, we gain having more detailed knowledge of the results, having more detailed, in-depth discussion of them. We also do all of this in very close collaboration with OECD. EFSA is chairing the OECD DNT in vitro battery expert group since 2018, and there are several ongoing activities under the WNT programs, but also under the Working Party for Hazard Assessment at OECD. I think there are 3 main concepts that we need to have in mind when we will hear the presentations of Brain Health Consortium. One is in vitro batteries of assays. We will be talking about developing methods based on in vitro. These methods measure biomarkers of early effects in vitro methods, in particular today. The aim of EFSA is to develop these assays in line with the OECD guidance 211 and in line with the top STEM criteria, allowing us to assess the readiness criteria to assess when is the most appropriate moment to continue for the regulatory implementation of those, or whether that is not the appropriate line to follow. We will also hear a lot about AOPs. AOPs are used in the EFSA neurotoxicity project as a framework to develop the assays, but also to interpret them, to interpret these biomarkers of early effects and link them to adverse outcomes. We will hear also about IATAS. IATAS is a framework developed by OECD in 2016, and it is nothing else than a way of doing a weight of evidence, but including also mechanistic evidence. So including all the available evidence from in vivo, in vitro, and human studies. I wanted to say that, of course, there is a lot of people also from the EFSA side supporting EFSA neurotoxicity projects. Here we have our collaborators also from ECA and GRC that help and support the EFSA neurotoxicity projects in general. And of course, Andrea Terron, that was one of the main driver of all of this work. And with this, I have the pleasure to give the floor and the computer to Ellen Frisch. Thank you, Ellen, for accepting having a presentation and informal talk with us on brain health. Ellen Frisch is a director of the Swiss Center for Applied Human Toxicology in Basel now, but she is one of the test developers of the DNT in vitro battery. I have a long history of developing in vitro methods for neurotoxicology. Thank you, Ellen, for accepting. It is a pleasure to have you here.
Well, thank you very much for your kind introduction, Iris, and it's my pleasure to be here and to introduce the session on the Brain Health Project today. Now, first, I give you a short background why we've been working on EFSA specifically has been working on developmental neurotoxicity, short DNT. So there is an increase in neurodevelopmental disorders in children. And the reasons are not really clear, but chemicals are suspected to contribute to this increase in disease. However, chemical DNT testing is not per se required in any regulation. And so the one issue is that the DNT guideline studies that are available right now from the OECD, they are really not suited, and they were per se not made for testing thousands of chemicals on their DNT potential. Moreover, there is a knowledge gap on the DNT potential of chemicals, and also because there are really high uncertainties hampering the application of regulatory DNT studies. So what is the solution for that? How can we close the current data gap? Well, the solution was the DNT in vitro battery that has been set up over many years in the past, and the test methods have been developed by 3 labs. It was the US EPA and mainly Tim Schafer here, University of Constance, and Marcel Leist is the PI there, and my former lab at the IOF. And so the DNT in vitro battery consists of 17 different assays, and these 17 assays, they cover 8 key neurodevelopmental processes that are indicated here. So proliferation, migration, differentiation into neurons and oligodendrocytes, apoptosis, neurite outgrowth, synaptogenesis, and neural network formation. And the assays in the battery are mainly human cell-based. So overall, one could say that this battery is really an EFSA success story, because EFSA has been guiding the process of development from the innovation phase through the availability of test systems, forming a strategic roadmap, method readiness understanding, fit for purpose validation until OECD recognition, which Iris already talked about, then also supporting the NAM availability by a CRO. And we are, I think, right shortly before legal implementation into the new plant protection product legislation. So, and how did EFSA support this whole process? I try to summarize this in this slide. Like Iris already mentioned, there was the scientific opinion from 2013 stating that we really need better methods to understand the DNT potential of plant protection products. And in 2014 then, and EFSA also participated here in this international stakeholder network, ISTNET DNT workshop for creating a developmental neurotoxicity testing roadmap for regulatory purposes. And in the same year, actually, we published a literature review, a systematic one for the availability of DNT method, which was really the basis for the further steps of the assembly of the battery. And after it was clear that there are methods out there that are suited to form a battery, then in the next steps, actually, there was a common EFSA OECD workshop in Brussels in 2016 and 2017. This was published in this meeting report stating that it would be very valuable to have a DNT in vitro battery to close the current data gap. And after that, EFSA actually started to fund the first testing in a DNT in vitro battery, at least on the European side. And it then summated in two external scientific reports. One was about the protocols and the data from the DNT in vitro battery testing in 2020, and the second one on the interpretation of these results in 2021. And this document served actually as the basis for the OECD initial recommendation document that was published in 2023. And so this document is really the foundation of going further into regulatory acceptance of the battery. However, there were also a few uncertainties of the DNT in vitro battery that were addressed in this document. So first of all, there are some common NAM in vitro battery or NAM uncertainties. For example, which species are you looking at? What about liver metabolism, which is of course not covered in brain cell NAMs, chemical domains, so the more the general ones. So then there are also the specific uncertainties like the lack of assays for critical key events like myelination or the lack of microglia, incomplete coverage of cell-cell interactions, but also uncertain about the neuronal subtypes that we have in the assays and the maturation stages. While for some of these uncertainties, the current NUMs need to be further developed. There are other ones that basically need to have data extracted. So in addition to these NUM uncertainties, there is also an uncertainty which is general for NUMs. It's the availability of adverse outcome pathways. And in this article, it was pointed out that there are gaps in the DNT AOPs, like in most other areas, like beyond skin sensitization. Now, how is brain health going to address these uncertainties? So basically, and I'm not going to go into this in detail because my colleagues will do this in the next talks, but in work package 1, the NAMs will be developed that need further development. In work package 2, through single cell and bulk transcriptomics, we will start addressing the data gaps on, for example, the neuronal subtypes. And then in work package 3, development and refinement of AOPs are being done. And I would like to give you a sneak preview into how single cell data can also support AOP development with the example of AOP 442, which is an EFSA AOP. It's called binding of compounds to voltage-gated sodium channels, how this can lead to DNT. And this AOP was recently endorsed by the WNT and OECD accepted. Well, and the main part of the AOP is depicted up here. So it's basically concerning the neuronal expression of voltage-gated sodium channels and how this can alter neurotransmission, neural network formation, and lead to cognitive function decrease. However, there is a second part of this AOP which concerns oligodendrocyte differentiation. And this is something that actually came up while testing in the DNT in vitro battery, and pointing out that also the oligodendrocyte lineage leading to possible hypomyelination is really an important key event that will be addressed also in the brain health project. However, so we have taken up at the SCAT this second line, this oligodendrocyte line of this AOP by Litvina Gerner, and this was her master's studies, and now she's a PhD student at the SCAT, and she performed a semi-systematic literature review on these key event relationships of this AOP, and also she browsed single-cell RNA-seq data on the genes coding for voltage-gated sodium channels with the University of California Santa Cruz Cell Browser. And I'll just give you a very small insight into her data. On the left side, you have oligodendrocyte lineage data, and on the right side, you have neuronal lineage data. And I think you can appreciate that when you look at up here, the second trimester data. So this is the fetal period that certain voltage-gated sodium channel genes are relatively high expressed, and actually these are only in oligodendrocyte precursor cells, not in mature oligodendrocytes. And so in the oligodendrocytes with increasing maturation, the channels are downregulated again, and this is different in neurons. And so this should show that MIE target expression beyond the neurons also in glia cells, and here oligodendrocyte precursor cells in human developing brains in vivo supports the human relevance actually of this AOP. Now, how is the battery going to be thought to be implemented? You all are aware of the Commission roadmap towards phasing out animal testing for chemical safety assessment, which will be published in the first quarter now of 2026. And the short-term action in the roadmap on the DNT in vitro battery are for reassessment of plant protection products, but may also be considered for human pharmaceuticals if needed. And in the medium-term action, the expansion also to reach relevant chemicals, which will be certainly also tonnage dependent, are envisioned. So, but what about the pesticides? So there is a suggested new annex of the plant protection product legislation, and I will give you just in brief the details which have been suggested by the European Commission, but it's not finally out yet. So the most important thing is that developmental neurotoxicity shall always be assessed for plant protection products, and that the DNT in vivo guideline testing can be waived even if the DNT in vitro battery is negative and the weight of evidence assessment has shown reliability and there is no other concern for DNT. Then when the DNT in vitro battery is positive, then you need further quantitative exposure considerations, but additional in vivo studies to investigate DNT shall only be undertaken when it is not possible to carry out a risk assessment based on all available information. So this is the provisional text, and I think this looks very promising, and I think we're all very excited to see the battery finally to be legally implemented. And what are the current activities supporting the regulatory implementation of the battery? And Iris also pointed out some of these activities. So for one, EFSA is currently sponsoring the lab-to-lab transfer of all the assays from the assay developers to the CRO DNTox, which will be not only to show that the assays are transferable, but they will also be available through DNTox. Secondly, EFSA is hosting the quantitative in vitro to in vivo extrapolation working group, IATA case studies, then the Brain Health Project, DNT in vitro to in vivo comparison, and preparing a list of DNT positive chemicals. Moreover, EFSA is also supporting the OECD and the DNT in vitro battery expert group, and which is also working on a tiered and defined approach. And moreover, the PARC project, which was already mentioned, is working on DNT AOPs in the new funding line starting from May, also new DNT case studies. And there is a new project initiated by ECHA with possibly providing a possible new hazard class for neurotoxicity. And the question is here how to delineate between adult and developmental neurotoxicity using NAMs. So this is also highly exciting. And with this, I'm at my end, and I acknowledge everybody who has been involved in the DNT in vitro battery development and acceptance. It's a huge international DNT consortium, And especially EFSA, I have to thank for this constant support, also the US EPA, the OECD, and then the Brain Health Partners. Thank you very much.
Thank you, Ellen, for this nice and comprehensive introduction to Brain Health Project, but also telling a bit about the history of why we are here today in EFSA. Thank you very much. There is some time for one or two questions from the room or online. If there are no, I can start. You mentioned in one of the slides that there is under discussion, I think, in the roadmap. We don't have here today our colleague in charge of the roadmap of the use of the DNT in vitro battery for other contexts of use in addition to pesticides. What is the current discussion on that area? I mean, because this perhaps could be of interest for our colleagues in the contaminants area or in the where they use other chemicals for example, food contact materials.
Yeah, this is a great question, Iris. So during the Human Health Working Group in preparation of the European roadmap, this was actually discussed. I mean, it came from EMA that they were thinking that it could be a context of use to use it also for assessing pharmaceuticals, but I have not followed up this, so I don't know where it's standing. And for the REACH relevant chemicals, well, I think the PARC project that I pointed out for introducing a possible new hazard class for neurotoxicity. I mean, this is also very promising. This will go over 3 years. And actually, we're leading this project together with Tamara Tal from the UFZ. And the idea is to generate really hypothesis how NAMs could delineate based on biology and neurotoxicity mode of action between DNT and ANT. And then if the data that is right there is not sufficient, we will also produce more data to either confirm the hypothesis or dismiss it. So this is actually the plan. So I think this is a great project and will bring us closer to also having REACH chemicals evaluated with neurotoxicity NAMs.
Thank you, Ellen. Yes, indeed. I think this ECAPARC project is very promising also to expand the use of the DNT in vitro battery for chemicals in general and the REACH. And I am sure I will discuss this with my ECA colleagues in the neurotoxicity working group. So I don't see more questions, and we are going to move to Otvar Mir. Otvar is the lead of Brain Health projects. He is coordinating all this amount of work in the different consortium project areas. He has more than 25 years of experience working in vitro and in vivo neurotoxicology and AOP development, and of course, in vitro methods development. Thank you, Otvar, for accepting joining this. I can share your slides if you prefer. You prefer to sit?
I can sit there.
Okay. It's fine. Of course.
Sweden logistics. Yes, I think it's fine. So thank you so much, Iris, for the kind introduction. And I would also like to thank the EFSA organizers for giving us this opportunity to present our project. So I will begin my presentation with a short background on our project entitled, as you see here, New Approach Methodologies, or NAMs, to study developmental glial cell toxicity of food toxicants, the Brain Health Project, EFSA funded. So this wonderful group of scientists makes up the Brain Health Project, coming from 4 different research organizations. And you will soon hear talks from Marie Gabrielle and Katarina Koch on the developments in work package 1, in addition to myself. Marcin will take us through the transcriptomics part of the project, and Anna will take us through the AOP developments. So this picture shows the overview of synergies with current international DNT projects where brain health scientists are involved. On the left side of this picture, you see activities from the ONTOX Task 91 project. This is headed by Ellen Fitche and her postdoc Eliska Kukowska. So the brain health project take advantage of activities here like brain physiological maps. Also, the DNT-AOP network has been built here. And indeed, characterization of the applicability domain of in vitro NAMs, including our synaptogenesis model. This has been very helpful for our brain health project because here we use this model to add co-culture with microglia. I will come back to this. On the right side, on the slide, you see the HARP project. Here we are about 15 partners, I think. So the aim here is to close uncertainty in the DNT in vitro battery and also to build an in vitro battery of odorous neurotoxicity. We are currently doing compound testing. You see a cut of a picture of a list here. This is done blinded. So this is tested across the NAMs for the partners in the park. And interestingly, there are 14 compounds we should share with the Brain Health Consortium from this list. Indeed, we are collaborating with the EFSA lot projects, exemplified here for lot 4, DNT RAP 4, pioneering non-animal method-based DNT risk assessment of 4 pesticides. This is coordinated by Martin Paparella. So the role of brain health here will be to feed the role of glia cells for DNT into an AOP-informed IATA case study on pesticides. This will be done in the end of the brain health project. So now I will dive into more details of what we're doing from the Norwegian Institute of Public Health side on the synaptogenesis model. So some background. Characterization and confirmation of the synaptogenesis assay has been described in the references to the left on this slide. The high content imaging pictures here are examples from our lab, where we see the neuronal marker MAP2 in green. You see the presynaptic marker synaptophysin or SIP in yellow, and to the right here we see the postsynaptic marker PSD95 in red. To the very left here we see a composite of these pictures, where we see the orange spots here indicate the overlap between the pre and post synapses. So, and this picture is from a recent paper in collaboration with the IF, where we did RNA-seq on the NAMs, undergoing differentiation in our case for 28 days. I also referred to this in the previous slide. I will not go into details here, but this picture shows the genes encoding pre and post synaptic expression, gene expression at all differentiation stages. In this case, day 3, 14, and 21 of neuronal differentiation. And as you see here in the red squares, you see the genes encoding the synaptophysin shown in the pictures above here, and also the LG4, which encodes the PSD95, as you see in the upper pictures. So, and you nicely see that the genes increase with the differentiation time here, and we like that. So this picture was also shown by Ellen in the previous presentation. So overall in this project is to study the astrocyte and microglia contribution to the synaptogenesis. In the current DNT in vitro battery, the synaptogenesis assay is based on rat cortical cell assay, where there is no astrocytes or microglia. So this is a new contribution to such an synaptogenesis assay. So how are we doing this? So this is an overview of the additional human inducible microglia, or IMG, to the synaptogenesis assay. So what we do is that we differentiate the synaptogenesis assay for 28 days, and after a few days of differentiation, we add the microglia. The readout is, as usual, maturation of synaptogenesis. And this is to study the important role of microglia to neurodevelopment. So we will do these assays with and without microglia. In the end of the project, in the coming 2 years, we will challenge these systems, including the synaptogenesis assay, with reference or chemical compounds, and we will then study the contributions of the astrocytes, but indeed the microglia, as exemplified there. So how do we do this co-culturing? This is a picture showing the refinement of the human synaptogenesis assay, the timelines here. You see the line here describes the differentiation days of the neuron astrocyte culture, going from day 0 to day 28, which is the end of the assay as we have defined it. So to find the optimal, the best co-culturing time point, we have added the microglia to the culture at different time or different days, which is day 3, 7, and 14 of neuronal differentiation. So this is a high-content imaging picture of the microglia monoculture, showing luckily that they are stained with the well-known microglia markers shown here in red. So since we succeeded in showing this, and also the other labs have done the same, we took these markers into the co-culturing, which is the aim of our part of the project. Here we see high-content imaging of the microglia co-culturing at day 3, 7, and 14. These pictures are taken at day 28 on neuronal differentiation. And this is exemplified with the EBO1 and the PU1 marker. So, but the message here is that this confirms the presence of microglia also at the day 28 on neuron differentiation, which we were happy to see. We also wanted to study if the microglia addition had an effect on the maturation of the neurons in the assay. So to do this, we again added the microglia at day 3, 7, and 14, and we studied the maturation markers, the progenitor cell marker nestin in the upper panel here. In the middle, the more immature neuron marker 2B3, and in the lower panel, we see the MAP2, which is a mature neuron marker. So this also confirms that neuronal maturation, even after 28 days of differentiations, looks promising, except for one observation here. If you look at the lower left picture here, you see some disturbance of the neurites after adding the microglia at day 3 on neuronal differentiation. So this indicates that if you add the microglia too early in the neuronal differentiation, this may lead to disturbance of the neurite maturation. And this is confirmed in this picture. So in the upper left part here, we see how the neurites look at, is looking at day 28 when the microglia are added at day 3. At quite early in the differentiation of neurons compared to the control to the right. In the lower panel, we see how the cultures look when adding the microglia at day 14 of differentiation. And here the culture looks quite similar to the control. So it looks more promising to merge the cultures at day 14 compared to day 3. Since we want to develop a synaptogenesis assay, we also wanted to study how microglia addition could affect the excitatory and inhibitory synapses shown by these markers here, the synaptophysin, VGAT, and VGLUT. VGAT is the inhibitory marker or GABAergic marker. And in summary, we see that this also supports the presence of mature excitatory and inhibitory synapses in the co-cultures at 20 days, 8 days of differentiation, which is good. So this is a bit technical slide. I will try to take you safely through it. So this shows quantification of neurite outgrowth at day 28 of neuronal differentiation. So what have we done here? So what we see is that in red bars, it's the neuron-only culture, and the blue ones are the co-culture with microglia. So in the upper part here, we see that additional, and these are done at day 28, as I said, but the merging of the co-culturing has been done on day 3, 7, and 14, as in the other pictures that I've shown. Upper panel shows that the additional microglia does not affect cell death. The total neurons count looks similar between the co-culture and the neuron control, and also the total cell count look good. In the mid panel here, we see that to the very left here, we see that the neurites count and also the Brown's point seems to go a little down in the co-culture when adding the microglia at day 3 and even at day 7. To the right here, we see that the neurite length seems to increase a little. So we need to repeat this more times perhaps, but our interpretation is that this support the previous picture, that the best time point of merging the microglia is later than 3 and 7 days. So it seems like the day 14 is the best time point for the co-culturing. We have also done a parallel gene expression study to support the high quantity imaging results. So this is very technical, and I will not dive into all the details here, but I will mention that the orange bars here are the neurites or the neuronal culture only, and the green is the co-culture. So here we have studied the microglia marker PU1 and CD11B. Yeah, the take-home message here is that the gene expression here confirmed the presence of microglia at neuronal differentiation day 28, also by gene markers. We have done the same for the astrocyte markers, S100 beta and aquaporin 4. This is just a selection of genes. We are currently studying more genes and more repetition of experiments. But this also quite nice to show that the gene expression describing, or the astrocyte gene expression markers increasingly expressed over neuronal differentiation time. And S100 beta and aquaporin P4 show highest expression and differentiation day 28, which is quite interesting. Moving on. We also wanted to check that the maturation of the neurons was not disturbed after co-culturing in the microglia. In the upper panel here, we see the expected decrease in the progenitor cell marker nestin, which was expected, and we like that. We don't see any differences between the neuron-only culture and the co-culture. So that was good to know. And in the lower panel, we see the synaptic marker synaptophysin. And we see a quite nice increase over differentiation time here. So this shows that the microglia co-culturing does not negatively affect maturation markers with differentiation time in the co-culture. So lastly, we wanted to study the postsynaptic markers. In this case, example, DLG4 encoding postsynaptic density protein 95 and SHANK3 show that and we see here that these markers increase over differentiation time, showing that the co-culture reaching a neuronal maturation over differentiation time. So again, the microglia addition does not negatively affect the postsynaptic markers with differentiation time in the co-culture compared to the neuron-only cultures. This is in line with the presynaptic marker in the previous week. To sum up, we have established the iPS-derived samptogenesis assay co-culture with the microglia that address uncertainties within the DNT in vitro battery related to the underrepresentation of glia-specific toxicities, neuroinflammation, and species differences. Yeah, and transcriptomics, Marcin will talk about this later, will be combined with the exposure of cell-specific model compounds and pesticides for test system characterization. In our case, maturation of samptogenesis readout. And as mentioned, we will also collaborate with the Lot 4 EFSA project on AOP in Formiata pesticide case study in collaboration with Martin. Thank you. That was, and I would of course like to indeed thank the Brain Health Consortium for the excellent collaboration. Thank you.
Thank you, Otmar. Thank you very much for this presentation. It's nice to see some of the results of the project already. A quick question from my side. So since now, based on the results, it is clear that so the days in vitro needed to have also glia have been already agreed. It is foreseen to start testing with chemicals in the next step, or what are the next steps in practice?
Exactly, yeah.
Thank you.
Yes, so we think we are about to be ready to start the testing of the selected chemicals for the project. So we hope to start that by late spring. I see nodding by my colleagues at the lab here. So yes, we think we are ready quite soon, and we also want to publish this characterization of the samptogenesis model, hopefully by spring.
So perhaps we can discuss this afternoon, which chemicals and how many can be tested based on the project budget.
Yes.
Thank you. Any question or comment? Also from the consortium, feel free to speak. Doesn't seem so. So we are going to move to the next speaker. It's my pleasure to introduce Katarina Coach. Katarina is also working with EFSA in other DNT in vitro battery projects. She's a junior group leader at the EU University, specifically in the Leibniz Research Institute for Environmental Medicine in Düsseldorf. She's also co-founder of DNTox, which is a CRO that provides services on the DNT in vitro battery and other neurotoxicity in vitro assays. Welcome, Katarina. Thank you for accepting discussing the current results of brain health of your work packages. She will be talking about addressing uncertainties in the DNT in vitro battery by incorporating glial cells.
Thank you very much, Iris, for the kind introduction and to be able to be here and present our data. The data I'm going to present today was initiated, or the work I'm going to present today was initiated by Ellen when she was still working at the IOF and is now continued in my working group since I took over 2 years ago. I have to declare conflict of interest. I'm a co-founder and shareholder of the DNTox, which offers testing in the DNT in vitro battery methods. However, what I'm going to present today is based completely on my personal views. As Otmar already mentioned, in brain health, we are trying to address uncertainties in the DNT in vitro battery. And the first one I want to talk about is actually due to species differences, because some assays are still based on rodent cells, and one is the neural network formation assay from the US EPA, which is based on primary red cells. And at the IOF, we have developed a human-based alternative, the human neural network formation assay. It is initially established on a commercial kit from the company NewSight, which is composed on a defined ratio of glutamatergic neurons, GABAergic neurons, and primary astrocytes. And these cells are plated on multi-electrode array plates, and electrical activity is measured over the time course of neural network formation. What we're doing currently in this work is performed by Ricardo Guzzo and Edda Zör, which are also here today. We are developing an in-house alternative to the commercial kit to be also independent from this provider. The assay is based on the following exposure scheme. The network is formed over 35 days. And starting on day 7, the network is exposed to the test chemical, and network activity is measured each week. We also perform a washout prior to electrical activity measurement to only look at chronic and not acute effects of the chemicals. In total, we are measuring 14 different parameters of the networks, including spikes, bursts, and network bursts. So not only network activity, but also maturation and synchronicity. Over the time course of the assay, the network matures, as you can see here in the spike raster plots. First, you have only a couple of spikes, and then when the network progresses or the maturation progresses, you see more spikes and also bursts. And when the network is completely matured, you can only see network bursts here in blue over the overall net electrodes of the well. We have tested several positive chemicals to confirm that the neuronal subtypes are also functional. What you can see here is exposure to a GABA A receptor antagonist, which, as expected, causes increase in network activity. And on the other side, I brought here data from an AMPA receptor antagonist, which, as expected, reduces the electrical activity of the network. In a previous project funded by the Danish EPA, we've tested several chemicals in this assay, mainly pesticides. And although this testing was very successful, uncertainties remained. And one uncertainty is that we are still dependent, or were dependent, on this supplier Nucide for the cells. But so far we have good results with our in-house developed cells. So this network maturation over the time course of the assay, you can see here, is based on our in-house generated IPS cells and our primary astrocytes we are culturing at the IOF. And you can see on the left that the network activity increases as expected over the assay duration, and on the right side you can see that the synchronicity increases as expected. Other uncertainties are that currently microglia are not present in the assay setup, and we do not fully understand the mode of action. We will also address this in brain health during transcriptomics. The second uncertainty in the battery is actually that we're not directly addressing astrocytes. So we have developed an assay for astrocyte development and function. And first, I want to talk about how we generate astrocytes in the lab. And this work is performed by Etta and Adi, who are also here today. And we have the opportunity to generate astrocytes from neural progenitor cells in 2 different ways. On the one hand, we can spontaneously differentiate them into a mixed culture of neurons and astrocytes. Or what we can do in the lower part, we can differentiate astrocytes in a enriched culture, almost pure astrocytes, in presence of BMP2 and CNTF. And when we look at the PCA, you can also see that these different cells cluster very nicely. On the top, you can see the NPCs, and on the bottom, you can see the matured astrocyte. On the right side, you can see the spontaneously differentiated astrocytes. What we did then is we characterized the astrocytes for their morphology, marker expression, but also functionality. On the one hand, we looked at the expression of astrocyte-specific markers. On the left side, you always see the spontaneously differentiated astrocytes, and on the right side, the matured astrocytes. You can see that they express S100 beta and aquaporine. But with regards to GFAP, which is a very mature marker, you can see that only the astrocytes exposed to BMP2 and CNTF upregulate GFAP expression. So we continued with this cell type or differentiation protocol to set up the assay. When we look at the transcriptomes of those astrocytes, I want to highlight here that they upregulate genes involved in signal transduction, nervous system development, and also inflammatory response, because it's important that they're also functional, regulating inflammatory processes. Staying with the transcriptome, we compared actually astrocyte-specific markers, both in the spontaneously differentiated astrocytes and matured astrocytes. Here you can see markers of astrocyte progenitors, so early genes, which are highly expressed in the spontaneously differentiated astrocytes and lower in the matured ones. And when we look at mature astrocyte marker, we were happy to see that these were low in the spontaneously differentiated astrocytes, but highly expressed in our matured astrocytes. So again, that brought us to the conclusion that we will stick to the protocol where we mature the astrocytes a bit more. Speaking of inflammatory competence, we assessed if how these matured astrocytes react to interferon alpha and interferon gamma and TNF alpha exposure. And we could see for TNF alpha on the bottom that concentration dependently they upregulate ICAM1, which is a marker for inflammatory competent astrocytes. And you can also see that they changed their stellate morphology to a very inflamed one. Last but not least, for the astrocytes, we also assess their functionality. They are normally actively regulating the glutamate concentration, taking up glutamate to control the concentrations available to the neurons. And here we developed this neurotransmitter glutamate assay, where we expose the astrocytes, or we don't expose the astrocytes, but we culture them in medium containing a defined concentration of glutamate, and we actually measure the uptake of glutamate from the medium by the astrocytes. And what you can see on the right is that with increasing time, the astrocytes take up glutamate, which means that you have decreased concentration of glutamate left in the medium. And when we inhibited the glutamate transporter EAT1 with UCPH101, we could see, and this is highlighted on the right side in orange, that the glutamate uptake is decreased, so more glutamate is remaining in the medium, which confirmed that the astrocytes are really actively taking up the glutamate. So based on these well-characterized astrocytes, we developed this assay, which takes 5 days, and the astrocytes are exposed to chemicals during this differentiation process for the total of 5 days. And at the end, we assess astrocyte-specific markers, GFAP. We assess astrocyte migration and also their astrocyte number, so the differentiation. I brought you one of the brain health chemicals because we already started chemical screening, and this is chlorpyrifos and we found that the most sensitive endpoint was actually the length of GFAP positive processes, which was concentration dependently reduced. You can see that here in purple, and also on the right side, I think can clearly see that their stellate morphology is not there anymore, and they lose their processes. It's also highlighted here in purple. Last point, we also want to address the microglia presence in our assays, and therefore we integrate microglia both in the astrocyte maturation assay and into the neural network formation assay. The microglia we are using have been generated at the IOF, and they overexpress CEBPA and SPL1. We have generated IPS lines both on female and male background, and generating microglia from these IPS cells. The protocol takes 9 days and uses different media. I'm not going into detail here, but at the end, we were able to characterize the microglia and confirmed expression of microglia-specific marker, which you can see on the right side, TMEM119, IBA1, and P2RY12. Last slide here. So we are integrating these microglia now into the NNF assay and also to the astrocytes. What we're doing for the NNF assay, we first seed the astrocytes, then add the neurons, and then after giving the neurons and astrocytes some time, we then add the microglia to the co-culture. We have here used two different strategies with regards to the maturity of the microglia. We either took microglia that only differentiated for 2 days, so very immature microglia, and integrated them into the co-culture. And here with the little arrows, you can see the microglia. We are currently optimizing the staining. They are not so easy to depict here. And what we're also doing is we take more mature microglia after 8 days of differentiation and also integrate them into the co-culture. What we're finalizing now is characterizing this co-culture, and then we are continuing with the chemical testing also for this assay. To sum up, we have established the NNF assay and the HEM assay, which are addressing two uncertainties in the DNT in vitro battery, which are glia cell-specific toxicity and species differences. And we are now performing transcriptomics to characterize these cells in more detail and also identify biomarkers. And we will, of course, then also continue with the pesticide testing and comparison to historical data in the DNT in vitro battery. With that, I want to thank not only my group, but also Ellen for starting this work and letting me continue with the work, and all the brain health partners, the whole consortium, for the continuous support and nice discussions. And of course, all funding bodies, and especially the EFSA, not only for the funding, but also for the support during our meetings. That's the end of my talk.
Thank you, Katarina. Very nice presentation. I'm excited to see a lot of new results since our last meeting. I have a couple of questions and comments. Since I don't see anybody, I'm going to jump. So I think this was a very nice presentation for us to see what comes before we have the in vitro NAM method comes to us. So all the work that comes before in order to establish technically the method in the say from the test system point of view. So super interesting. Then I think it's also a very challenging part of the project, because here we are in a situation in which we have the DNT in vitro battery that has been considered sufficiently good to be used in a specific context of use or for specific uses. And this is, of course, from the scientifically and technical point of view, an improvement of that. But from the regulatory point of view, we are in the situation that it has to show that it's an added value for a regulator. That for us is the final number that is going to give us in the risk assessment. And then, because it's clear when you present that the added value from the scientific point of view is there. So something to think together how this should be done, in the sense that how many substances should be compared with the old test system and the new test system, what are the best ones to select, and these kind of questions that we need to address. I think also it was very interesting, the transcriptomic part. Maybe you can explain a bit more what is the purpose on the transcriptomic, if it's only to characterize molecularly the cells, or if it's also to be used with chemicals, so for us in an AOP-driven context. Would you explain more about that?
Yes.
Thank you.
Initially, it was performed really to characterize the cells, because we wanted to find out if there's a difference between those two protocols, with the astrocyte maturation and with this spontaneous differentiation. And we also did transcriptomics for the TNF alpha exposure and interferon gamma exposure. And this was very interesting. I didn't have time to show this because they generate a completely different transcriptomic pattern, these two inflammatory molecules. So we could, of course, think of extracting a little panel of genes. So not a whole transcriptome panel, but maybe some certain genes, which then could be function as transcriptomic readouts for certain inflammatory processes being activated in the astrocytes. So we really think that based on the inflammatory stimuli, we generate different subtypes of astrocytes or activate different subtypes of astrocytes. So this could be something that could be done in the future. So we are, or ETA is mainly looking at the specifics of that, because all this transcriptome data gives us so many information. So we're trying to find out which genes are most relevant when activated by the different NOXA and yeah, so it was basically first as a characterization intent, but it can also be used to generate panels which can be used after chemical exposure to see which inflammatory processes had been activated, for example.
So yeah, I have a question, maybe more down, not so academic, but again back to the practicalities. So because you've tested the Oxon, right?
The Oxon, yes.
Yeah.
Mm-hmm.
So the problem we have is that it's all fine to have this. I'm going to be blunt, to have, and I've been asking this question for years. Mm-hmm. It's all fine to have the testing, but if you don't know the toxic moiety, be it the parent compound or the metabolite, what we're looking at is completely wrong. Mm-hmm. So the problem is you need to have the metabolite available to be tested. It has to be synthesized, and you need analytical methods that is relevant to that. Because if we want to do, we've published this PBPK document to interpret quantitatively, to eventually calculate safe levels based on these. If we don't have that, we can't do it. Yeah. As simple as that. So what do you think?
Yeah, that's definitely a limitation, that our cells are to a certain extent capable of metabolizing chemicals, but of course not as the liver would be. So we are not sure if the chemicals can generate metabolites that could be more toxic than the parent compound. So if there are metabolites available, we could test. It makes maybe sense to test both the parent compound and the metabolite. But if the metabolite is not available, this is definitely a limitation.
Wow, that's big, because you know that's going to be the elephant in the room. And very often, I don't know if you've seen this, but with the first AppCrack case study with Katie Paul Friedman, published in 2020, we looked at 448 compounds, and we tried to derive points of departure based on, you know, HTTK kind of IVV tier 0 type approach with Cmax predictions versus the no-AL, low-AL in animals. Most of the time, 89% of the time, the POD derived on an AM basis is more conservative than the traditional one based on animals, except when the compound is bioactivated by metabolism, organophosphates, carbamates, you name it, systematically. So I think that's going to be maybe something to handle, because otherwise, what we, you know, this question will always come around. I don't know if, so the idea is, you know, at the OECD level, they are now developing testing methods for very simple stuff to derive CLINTs, you know, intrinsic clearance, fraction unbound of the compound, gastrointestinal absorption, and so on and so on, to then parameterize those. But again, if we don't have information on the metabolites that are generated and the consequence of that, maybe with QSAR we can actually see if the compound is bioactivated. I think that's pretty much possible these days.
To get the information, but of course it won't be possible for all chemicals.
Yeah. I mean, one of the things we're trying to explore now, so we have generic models that are available for parent compounds, so generic model for rats, humans, and farm animals. What we're trying to do is to introduce metabolites generically in the model as well. That's already available in PKSIM, but of course, if we don't have the metabolite, then we can make an assumption, but then, because that's the only way to translate that back to, you know, blood concentration in the brain, blood concentration or brain concentrations.
Yeah. And the issue is also that metabolism in the rat could be completely different from metabolism in the human. So it doesn't tell you 100% that when you see a certain metabolite in the rat, it must be the same in the human. So it's not always.
Well, since 2013, the dossiers now, they have to submit comparative metabolism. You know, we published that document in 2021. So comparative metabolism between humans and rats or test species to look at disproportionate metabolites. So part of that is handled already. Okay, sorry. Yeah.
No, thank you, Jean-Luc. I think that's a super important topic when we talk of using NAMs for risk assessment. And indeed, it will depend on the context of use, because, for example, in pesticides, we have a very good overview of the metabolites in rat because we have the ACME study, and we have also the in vitro comparison in different species that allow us to know which are the metabolites to be tested, and it could be synthesized and tested in vitro. However, in other areas, I think the data that you have from the ADME point of view is more limited than in pesticides.
And of course, these are not metabolically competent, right?
To a certain extent, they are, but
Okay.
but not as the liver.
Yeah,
So
sure.
of course, they don't have all the SIPs.
I think there are a lot of work ongoing on on increasing, from one side, increasing the metabolic competence of the DNT in vitro battery by adding specific things, and from the other side to understand what is the metabolic competence. None of them are part of the IRMA projects we are discussing today. There is a lot ongoing. Probably you are more aware than us, Jean-Luc. But indeed, this is a very important topic to continue discussing. So I think I'm going to move. Thank you, Katarina, for the presentation. And I have the pleasure to welcome to Marie-Gabrielle Zurich. She is the manager of one of the work packages of brain health. She's a group leader of the University of Lausanne, also long-standing experience as neurotoxicology and in particular developmental neurotoxicity. And she's specialized in glial cells and cell-cells interaction. She's gonna present to us whether can we not quantify the myelination in vitro. Just as a background, myelination, it is of course one of the key neurodevelopmental processes needed during the development of the nervous system. And in the OECD DNT in vitro battery recommendation document, it is noted as one of the gaps needed to be fulfilled to have the full overview of key neurodevelopmental processes in the battery. And Marie-Gabrielle is working with her team very hard to do that. Thank you.
Okay, thank you, Iris, for the invitation to be here. And yes, okay, so you introduced very well my talk because you mentioned that the myelination is a gap or an uncertainty in the DNT in vitro battery. So we are going to see now how we could quantify myelination or demyelination in vitro. So what is myelination? It's very basic, but I want to give a definition here so we are all on the same page. So myelination is a very highly and tightly regulated developmental process. It is regulated in space and time. And during this process, the oligodendrocytes are wrapping the axon with their membranes, which is lipid-rich, and these form the myelin sheath. And the myelin sheath allows a fast and rapid propagation of the action potential along the axon. So if you look at the so it's not moving? No. Okay. If you look at the picture on the right, so you see in yellow a neurons, and you see that the violet cells are oligodendrocytes that are wrapping their membrane around the axon. And if we make a transversal section, we will see inside the axon and outside the various layers of these membranes of the oligodendrocytes that are accumulating around the axon, and the entire violet thing that you are seeing here is the myelin. So if we consider the oligodendroglial lineage, we see that the cells are going through at least four major stages of differentiation. At first, you have the OPC, that are the oligodendrocyte precursor cells, and at the end of the maturation of this lineage, we find the oligodendrocytes that are called myelinating oligodendrocytes, and you see that these cells have a lot of processes, and at the end of these processes, there is like a kind of a flat area that is really made of membranes only, and this is this flat area that is wrapping around the axon and that gives the myelin. So to have myelin, to have the formation of the myelin around the axon, we need a close contact between the neurons and the oligodendrocyte. So how could we mimic this quite complex process in vitro. We decided to use a complex, clearly we need a complex system processing at least oligodendrocytes and neurons. So we decided to follow the protocol published by Pamies and al. In 2017, where they show how to produce spheroids, brain spheres, from human-induced pluripotent stem cells. So we go through various steps of differentiation, and we obtain at the end a brain sphere. Here, for example, each brain sphere will comprise neurons, astrocytes, and oligodendrocytes. And these structures, these spheroids, are showing spontaneous electrical activity that can be recorded. And the author showed at the time myelin-like structure. So, this is electron microscopy, and the black area here is very much looking as myelin in vivo. So, but we started to generate spheroids with the IMR90 IPS line that we are supposed to use in the Brain Health Consortium, but we couldn't reproduce these results. And furthermore, I want to point out on the fact that we need 8 weeks in culture to, the author here needed 8 weeks to generate this type of myelin-like structure. So it's quite long for an assay to be introduced in the DNT-IVB. So we decided to jump on another type of IPS line, the SON HIPSCs. SON is coming from the name of the 3 transcription factors that are overexpressed in this cell line. So we got these cells from Moritz Rosner in München, and thanks to the help of Katharina Koch. So this IPS line is very easily differentiated into oligodendrocytes in 2D. And you can see here the MBP staining. MBP stands for myelin basic protein. It is a protein that is very specific for oligodendrocytes and that is very enriched in the myelin sheath. So here it's a 2D preparation of the cells, and we can obtain oligodendrocyte in 12 days, which is really short time because normally it takes weeks. So we were very happy to see that, and then we started to generate spheroids from this IPS line. And we succeeded to prepare them, and then we compared these spheroids prepared from the SON IPS line, and you see the result in green on the graphs. We compared them to spheres prepared from the IMR90 that we were supposed to use in the project. And you can appreciate that the expression, the mRNA expression quantified by RT-qPCR for MBP is much, much higher in the spheres generated from the SON cells compared to the one generated from the IMR90. And more importantly, you can see that we can measure the MBP protein expression by ELISA in the green column, so the spheres generated from the SON cells, whereas, as I said before, we couldn't see any expression of MBP in the IMR90 derived brain sphere. And again, on the right part of the slide, you can see MBP immunostaining, and you can see very nice oligodendrocytes with nice processes that are stained for MBP in the brain spheres derived from SON, but nothing in the other brain spheres derived from the IMR90. And I want also to point on the fact that the MBP immunostaining was present already starting after at week 4. So we thought, okay, maybe we could reduce the time in culture for the assay. So after having observed and being able to quantify MBP, we exposed the spheres to non-demyelinating agent, so cuprison and lysophosphatidylcholine in green here. And you can see that the MBP protein level measured by ELISA is decreasing after cuprison exposure. That was, I forgot to say, during 1 week, repeated exposure during 1 week. And it's the same for the lysophosphatidylcholine in green. And this is observed in total absence of effect on cell viability, as you can see in the lower graphs. On the top of it, we have used ibuprofen as a negative control, and you can see that ibuprofen had no effect on viability neither, but also had no effect on the MBP protein level. So we were extremely happy with these results that we need to confirm. But then we thought, okay, we have MBP and we can decrease the MBP amount with demyelinating agent. Does this mean that we really have the presence of myelin in these spheres? So we started the analysis with electron microscopy, and you can see on the bottom pictures that we have structures that are resembling what was published and what is published for in vivo. So, but also we observed what is shown on the upper part of the figure. We can observe also layers of membranes that are less compacted, as here. And we can also observe in the first one less layers. So we are wondering, and we are hypothesizing, that this is the very first step of myelination. But we are not sure of that, and we need to investigate that better. So we are doing two things now. Sectional, how do we say that? Slices, like consecutive slices and 3D reconstruction by EM. And I had a video, but it's not working, so I cannot show you. And we can see with this type of reconstruction, we can appreciate that this type of structure is really going along some kind of structure that could be an axon. So we are more confident that maybe this is real myelin, but we need really to do that more extensively, and we need to perform immunogold labeling. So it means stain for MBP on the top of this structure. And if we can show the presence of MBP in this black structure, we would be extremely confident that this is really myelin. So I want just to show you that there are several publications out there claiming that they have myelin and in vitro. So this could be true. But I think for the moment, it's difficult to be sure of this claim, about this claim, because the authors are not showing much. They are showing only one picture by electron microscopy, and they are not proving that this is really containing MBP or other very specific protein for the myelin sheath. So, and I want to insist on the following fact. Most of the people show one picture by electron microscopy, and then they jump to looking at MBP immunostaining or quantification or another type of protein in their culture. And then instead of speaking about decreasing of MBP amount, they directly speak about demyelination. But we are not extremely sure, because in vivo, there is a parallel between decrease in MBP content and demyelination. This is clear. But when we started in vitro, we haven't tried to really characterize and make the parallel between the decrease in MBP content and the decrease of this kind of structure. So I'm quite confident that this is myelin, but it's not totally proven yet, I would say. So then I will go very fast on, we are also trying to add microglial cells in our model. Why? Because we want to assess the difference it makes to have them around in the outcome of in the neurotoxic outcome after exposure to chemical. But also it's important to have microglia in this system because it is known from the literature that they interact very strongly with oligodendrocytes and they contribute to myelination. So briefly, we added the iPS-derived microglia, the same cell as presented by Katarina before. We added them to the spheres, so either at the very start of the culture or after the spheres are formed and already quite differentiated. And in both cases, we can observe a TREM2 positive immunostaining after 8 weeks in culture. So TREM2 is very specific for microglia. So we were quite pleased to see that for the top part of the figure, the microglia that we introduced at the very beginning are still present after 8 weeks in cultures because when they are culture alone, they don't survive so well. So they need to have contact with, to be in contact with other cell types. So I want now to conclude. So in vitro myelination, we think that in vitro myelination needs proper characterization. And I insist that it has never been done for in vitro models. We think that the spheres derived from sun cells reproduce the very first step of myelination. However, we need a bit more characterization about that. And then we need to know something about the relationship between MBP amount and demyelination. So to make sure that MBP is really showing demyelination and not just, an effect on highly mature oligodendrocytes. And then last conclusion, IMG, as I said before, have been successfully incorporated in brain spheres. Of course, we need to test their functionality in the sphere. So a lot more work to perform. And very last conclusion, these results are extremely encouraging, I think. And EFSA funding is really critically important for that. So it's thanks to this funding that we have been able to make this important work and that we will be able to develop further this essay to quantify demyelination. With that, I'm done and I thank you for listening. Okay.
Thank you, Marie-Gabrielle. Thank you very much for this nice presentation and overview on where we are with the myelination assay to be incorporated in the DNT in vitro battery. I think this was also a very good example of what goes before the in vitro method is developed and all the steps for molecular, technical, and functional characterization. I think you showed two things that we are very used in the pesticides peer review. One is I have the paper. I want to assess whether it's not reliable. I see there is only one figure. I think we can relate to that feeling, and I like it also a lot that you clearly and transparently presented that you could not reproduce some of the previous results from other publications. That is also something we know it happens, and that is why it's so important, transferability of the methods and reproducibility of the results. I think the results are very optimistic because also in 6 months I didn't see anything, and I have this nice feeling on we are going to have very interesting results as part of Brain Health Project in the next 2 years. So thank you very much for that. I don't know if there is any comment, question. Doesn't seem so. So we are going to move to the next speaker. I think we're on time. I have the pleasure to introduce Anna Forsby from the Stockholm. Where is Anna? I'm here. But Oh, sorry, Marcin. Very bad chair. Forgot. I have the pleasure to introduce Marcin. I'm not going to say the surname because it's impossible for me. He's a senior researcher from the Norwegian Institute of Public Health, and he comes from a different area. He's a system biologist with expertise on omics, and he's trying to bring this expertise to the neurotoxicology as part of the project. He's managing this work package that is focuses on transcriptomic. Thank you, Marcin.
Thank you very much for the kind introduction and invitation for this meeting. I would like to give a brief overview of how we are going to implement the transcriptomics into glial signatures. But in order to do so, I should ask the question, why do we really want to do that? And for this forum, I think it's very important to mention that the adverse outcome pathway concept is extremely interesting concept, not only because it simplifies super complicated biology mechanisms, and it's a little bit like being a lawyer. So within this simple acyclic graph, so acyclic means one directional graph, it embeds the mechanistic understanding of the processes and simplifies them in such a way that within such a graph you have the most important knowledge that is necessary to give answer for the questions that regulatory people ask. What's the effect? Does the chemical A cause adverse outcome? So as you can see within this graph, the blue dots are representing our genes. So this is ambitious of the Brain Health Project to implement those transcriptomics signatures within the key events. If we succeed or not, the time will show, but I'm optimistic about that, seeing the development of the models. And within those AOPs is a knowledge which we gather within AOP Wiki, and those in the knowledge within the consortium, I think is pretty well developed for the DNTs. But in the Brain Health, we started in the parallel directions. So one of the things we actually stepped back a little bit and we looked into the gene ontologies, which contains the knowledge, transcriptomic knowledge on the different processes. And we challenged our consortiums, our experts, to pick up all of those gene ontology terms that were relevant for the developmental, for the models that we wanted to study. And we basically organized the voting. So from those, we were able to pick the correct gene ontologies, focusing on the development parts. And further on, from those relevant gene ontologies, we were able to focus on specific genes that are behind those gene terms. And maybe you can think that this is a trivial exercise, but actually it doesn't really show, like, because there is a lot of, we are still discussing, even the experts discuss what is the most important. And when we compare the knowledge also with the knowledge from, for example, from the single transcriptomics data, we generate a little bit the different knowledge as well. So we should also have that in mind. And with this exercise, we also, as a part of the project, we want to be transparent, and we develop the plans where it shows us where the data residues, where we can find the data, and where we will find the future and the data that we produce. Oops. The data management plan is one of the ambitions for the Brain Health here. So you can see basically what is right now in our service in Oslo. Because we are talking about the systems that are created in the lab, and they are not really taken from the developing brain per se, we then use the transcriptomics data available for us. And here we are talking about one of the model which was giving, which IUF and IPH did sequencing, and we were able to see that indeed 90%, 95% of those markers were present in these models. So this is a good piece of the information for us to start. Further on, in the brain health, we don't have yet data for the single cells transcriptomics, but we have been able to explore available data sets from the UNIL and that on the brain spheres. And Arup did great jobs on analyzing those markers and finding out we have some precursors of the neuronal markers, oligodendrocytes and astrocyte markers, but we have also found out challenges in terms of that maybe those data sets are not perfect, and we need to do better in the brain health project. But if you look at this particular graph, I would like to maybe not go to the details here, but I would like to mention that when we connect the genes with the AOPs and the different models, we have multiple systems biology approaches. We don't really have one single approach, but we want to combine the knowledge which we can find from the different databases. So we are using the available tools which are out there, but we are also actively developing the tools that we are using in the brain health, and they will be also available for another stakeholders to get a little bit closer to the real data. And here I will mention very briefly about the AOP Network Finder, and I will mention about AOP Enrich that we are developing in NIPH. But the idea for the brain health is that once we have the models with the microglia, we will do the single cell transcriptomics assays, and we will also have the compounds testing. So we will be delivering a lot of transcriptomics data with the as those response, and we will correlate them using network analysis with a real outcome. So here from high content imaging. And we know that we have to be extremely careful because we are looking at the developmental brains. We have to be extremely careful with the dynamics of those markers. And although we have published and see that those markers are changing, we don't really have yet the complete pictures of the models within the brain health when the microglia will be added. So this data is basically not produced yet. And but for the AOPs, and I mentioned AOP Wiki, the knowledge that the brain health will gather will be also deposited there. And so this is the work on the VP3 that Anna will present. But throughout the AOP Network Finder, we already facilitated the reconstruction of the networks from the different AOPs, visualization of the networks, and exporting the data for further exploration. And this is important because this is the obviously dynamic process. So in 2 years from now, we will be a little bit in the different situation that we are today, where we generate those. Maybe there will be someone else who will pop up the new VNTs, and with the AOP Network Finder, we will be able to pick those up extremely quickly and update our knowledge. And not only us, but also outside of the consortium. So some of the people who are online can go to this tool and have a look at how it works. When it comes to the linking the transcriptomic signatures to the IOPs, we also have to think in the novel ways. So we believe that if we will find out the transcriptomic signatures about given key events, we are able to create the tool which will be similar to the gene set enrichment analysis. So having the differential expressed genes, so in the context, for instance, of the chemical exposure, we will be able to use those differential expressed genes and put them into the tool, which will show us which of the key events are being upregulated within given dose. And as experts, then or regulatory people will be able to pick up which actually AOPs are here overrepresented, and so this is direct link with the regulatory work. So we have tested this only right now on signatures where we compared the day 14 and day 3, and you can see that the top key events are related to imperaxonal transporter reduction, neuronal synaptic inhibitions, neuronal network function increase. And here we have to stop for the second, because you have to remember that here we are just only looking at the development. So this is this extra difficulties where we are looking at the developing system. So if we want to apply the chemicals for the DNT, we all the time have to think about at which time points we are actually looking in our system. So I will maybe left this graph to Anna to discuss it a bit later on, but this is how we used AOP Network Finder right now to generate get the AOPs, find out the genes which people were describing, and we are curating them further on for our purpose. So with this, I would like to conclude that right now we have established the set of the genes markers within our consortium, but we have not stopped there. And of course, we are looking at all of the literature around us. We have made well-designed workflows for the transcriptomes, both for single cells and bulk analysis in our servers in Norway, and we are ready to apply them for the all transcriptomics data that will arriving from brain health from the different partners, and we have adopted FAIR principles for the comprehensive data management that will work behind the brain health as well. And so with that, we are ready for the cool things. And I would like to thank all of the brain health people here present and collaborators, and Arup for wonderful work on the transcriptomics data, and master students that work hardly on the developing the tools that are bringing closer and the data to the people who are not computing scientists. Thank you very much.
Thank you, Marcin, for this nice overview of the transcriptomic work package. I have to say that this is coordinated by my colleague, Giovanni Giacomo. He cannot be here this morning. He will be with us this afternoon. I'm sure he has more technical questions to your data. From my side, I think you say something very important for us, that is the FAIR principle and the data management. And we have discussed it a lot in brain health meetings, how to give us the appropriate raw data, how to have the image of the high content image, for example, methods, and also for the transcriptomic data. So we are sure that with the knowledge that is built with brain health project, if cannot be reproduced, we will know why having that data. So thank you for being always very, very positive and active with this, with us. And I think now we go with Anna. Yes. Otherwise, we won't have time. So Anna Forsby is the last speaker of today. She's a professor in the University of Stockholm in the Department of Biochemistry and Biophysics. She's also a neurotoxicologist with expertise on AOP and in vitro methods, and she's coordinating the work package of brain health, aiming to develop an DNT AOP network. Thank you, Anna.
Thank you so much, Iris. I'm super happy to be here and to meet all of you in 3D. So I will talk a little bit about how we can integrate the glial cell-related key events into DNT AOPs. And one of our tasks in our work package was to review all AOPs in AOP Wiki, or not all AOPs, all DNT-related AOPs in AOP Wiki. And there are now about 34 AOPs that are mentioned in Wiki that are related to developmental neurotoxicity. And some of the AOPs are endorsed, but others are very premature or under development. So 12 out of these 34 AUPs, they contain key events that are associated to radioglia, astrocytes or microglia, or oligodendrocytes. And the diverse outcomes for these AUPs containing glial cell-related events are impaired learning and memory, cognitive function decreased, and decreased IQ, impaired locomotor function, and neurodegeneration. So the DNT AUPs with glial cell-related key events, there is one concerning radioglia, and I think it was developed by Ellen and colleagues. It contains key events that regard disturbed radioglia alignment and migration, decreased radioglia cell density. There are 5 AUPs concerning astrocytes and microglia, and the key events, the glial cell-related key events are activation of microglia, increased pro-inflammatory mediators, and neuroinflammation. And then there are 5 AUPs concerning oligodendrocytes, and quite a lot of key events. I will not go through them. So there are some, or actually a lot of uncertainties and knowledge gaps in these DNT AUPs with relation to glial cells. So most of the AUPs are premature and under development. So there is a presentation of the graphical outline of the AUP, but without any supporting text, no abstract, no key event relationships, and nothing about how the key events can be measured, or no stressors or so, and maybe not even any references. So there's a lot of information lacking. No DNT AUP spells out astrocytes or astrocyte-specific events. So astrocytes can only be associated with inflammation, neuroinflammation, which can also be related to microglia. And most molecular knitting events concern hormone receptor interference. It could be agonism or antagonism. And it's also, we can also see that there's some important processes that involve glial cells that are missing in AOP Wiki. So also as a task in our work package was to perform a systematic literature review to identify glial cell related DNT key events that can be associated with exposure to food contaminants. So we did this, and we retrieved about 50 articles that for each cell type, which qualified in the appraisal processes. And in these 50 articles per cell type, we could identify hundreds of putative key events, or let's say events. So for clarity here, we focused on the events that were cited at least in at least 4 different articles. And we identified for radioglia, we identified novel key events that were decreased radioglia proliferation and increased the NPC differentiation to astrocytes. So the identified knowledge gap and understudied features for radioglia was radioglia proliferation, regulation of neurostem cell and neuroprogenitor cell differentiation into neurons, astrocytes, and oligodendrocytes. That this is what is called the neuroglial switch. For astrocytes, the novel events that were identified were astrocyte activation and a lot of astrocyte features. I will not mention all of them, but also the role of astrocytes for synaptogenesis. So what is understudied, the summary of understudied features, is astrocyte differentiation and astrocyte function, and astrocyte activation, and also the role of astrocytes for synapse formation and function. These are not identified in cited at least 4 times, but we also realized that
the role of astrocytes for neural network function and formation and function, and the role of astrocytes for blood-brain barrier function are missing in AUP wiki and are of course super important for neurodevelopment. And for oligodendrocytes, there are very many key events measured. I think that the most essential oligodendrocyte related key events are mentioned in AUP wiki. However, the relation between oligodendrocyte development and function and microglia and oxidative stress is not mentioned in AUP wiki, but identified in the literature. Also, impact on oligodendrocytes and aberrant synaptic formation and plasticity. So we have identified uncertainties and understudied features are synapse formation and function, neuroinflammation, axon formation and maintenance and neural network formation and function. So I really believe that, I mean, my partners are on the right track developing these assays. So what do we do with this information? One other task in our work package is to develop new AUPs. We tried a new approach here, maybe to us a new approach, and that is to use AI tools to see if how they perform in the AUP development. So we took one frequently mentioned event, that radioglia proliferation, decreased radioglia proliferation, and it was mentioned at least 4 times, and we call it a nodal key event. So 5 articles with this nodal key event were identified and uploaded into 2 AI tools. We used ChatGPT and Perplexity to see how these tools could perform in AUP development. And of course, the prompt is super important for the outcome. And here is one piece of information that was used as a prompt, and it's super important, of course, to instruct the target cells. Also, that MIEs, key events, and AOs should be identified. It should be developmental neurotoxicity. What we also realized that we have to specify that the information has to come from the result section, not the discussion section, because otherwise we will have misleading information in the AUP. And also very important is that the OECD guidelines for AUP construction should be followed. So we did this, and we were not very surprised. We were very happy when we saw the results, because I will not show everything. We got like a 50-page dossier from Perplexity. But the simplified linear DNT AUPs of these 5 articles showed that both ChatGPT and Perplexity could give or suggest AUPs that really make sense. So the molecular initiating events were, of course, different in the 5 articles, but they all concerned radioglia signaling pathways, which will affect radioglia proliferation in both. In the articles, there were also premature neuronal differentiation. There was astrogenic shift to also enhance gliogenesis. There was a depleted pool of progenitor cells also in both. There is impaired neurogenesis, and also the adverse outcomes were suggested or identified as neurodevelopmental impairment and cognitive deficits, and in both suggested by both AI tools. So I really think that there will be something that we can use from this, because it also speeds up the process extremely fast. So the conclusions of the work so far is that the review of the DNT AOPs in APO Wiki showed that the majority of DNT AOPs with glial cell-related key events, especially for oligodendrocytes, they're premature and need supporting information. So we can also help with that. And astrocytes could only be mentioned as regarding neuroinflammation, but astrocytes have, of course, many other functions in brain. And the literature review identified uncertainties that retroglia proliferation, regulation, or differentiation are significant events in DNT. We found novel key events regarding astrocyte differentiation, morphology, and activation and function. And also we saw that key events that describe the role of glial cells for neuronal synapse and neuronal network function. And the AI tools, we think that they will be very useful, both for data extraction from all the literature, and also to suggest putative AOPs. The time that we save on this is extremely, I mean, it took 15 minutes and then it was done. Of course, we have to manually curate it, but it would definitely speed up the process. So we use the approach to identify nodal key events for identification of significant articles that can be included in the same AOPs. Okay, so just the final slide here on how transcriptomics will be useful for, we will work on it in the next step, and also Ellen's example showed that transcriptomics will be very useful to support AOPs, and also, of course, the work in work package 2. So we work closely together. So we have focused here on the key events in existing AOPs and also in putative AOPs. These key events can be supported by transcriptomic profiles. So, of course, gene expression will contribute to the key events, but also by looking at transcriptomic profiles, we can identify new biological pathways that are not yet mentioned as key events, and then they have to be proven, of course, and there is like a circular interaction between these three parts. So I want to thank all of you guys, the Brain Health Consortium, EFSA, of course, who gives the possibility for us to do this. And then we have Vaya, who is in the plane from India now. He's sorry that he couldn't be here. He is the AOP expert here, and then Rosalind is also working a lot in this project. Nefeli did a lot of the astrocyte literature search, and Claudius did a lot of the oligodendrocyte literature search. So thank you very much.
Thank you, Anna, very much for this overview of where we are with the AOP development. I have some questions and comments, but since I promised we will finish at 12, I will do them in this afternoon meeting. I think it's very promising, all the knowledge that you are providing on DNT AOPs. And I think we need to close. Jean-Luc, from my side, I thank you very much to all for sharing all the knowledge and all the data you have so far in the middle of the project of brain health. I think I go home with a feeling of optimism. We say several times optimism, but also excitement of see what will happen in the next 2 years of the projects. I'm very happy that you mentioned that brain health is doing all of this work in collaboration with other EU projects. We hear EFSA project, but also PARC, we hear ECA, OECD. So I have the feeling that you are doing the best available science to assure that some of the uncertainties in the DNT in vitro battery perhaps can be addressed in the next 2 years. Perhaps we will need more time. We'll see. So thank you very much to all from my side.
Thank you very much.